Welcome to Ultimate Sleep Doctor. Tonight, we'll journey into a realm of astonishing secrets. Within the very blueprint of life, nestled within our DNA, lies a remarkable method of storage. Consider the immense capacity of a computer holding countless files. Now, picture that same kind of information encoded within the elegant spirals of our double helix. It's a beautiful, intricate system, a silent library of potential. This isn't just biology, it's data, patiently preserved, echoing the fundamental order of the universe. Let's explore this fascinating connection with a calm and curious mind. If you find these explorations soothing, please consider liking this video and subscribing to Ultimate Sleep Doctor. Together, we can soothe more wandering minds, one peaceful night at a time. Just breathe, let your thoughts drift like clouds across a twilight sky. Let's begin. Let's consider the incredible potential of DNA as a way to store information. At its heart, everything digital, your phone, your computer, relies on a simple concept: the bit. A bit is the smallest unit of information a computer can handle, and it can be either a 0 or a 1. Now, DNA, this remarkable molecule, already possesses a natural way to represent these binary digits. It uses just 4 building blocks: adenine A, thymine T, guanine G, and cytosine C. These are arranged in pairs. Adenine always joins with thymine, and guanine always joins with cytosine. We call these pairings base pairs. This AT and GC pairing is the fundamental code we'll use to store our data. To represent a 1, we could encode it as an AT base pair and a 0 as a GC base pair. It's a surprisingly elegant system, utilizing the inherent structure of DNA itself. Let's think about the physical dimensions involved. A single base pair, the distance between an A and a T, is approximately 0.36 nanometers. That's incredibly small, about 1,000th of the width of a human hair. Now, to read this information, we need a space around each base pair, a little gap for the machinery to access it accurately. Let's assume a spacing of 1 nanometer, a reasonable estimate for the machinery involved in reading the DNA. With a 0.36 nanometer base pair and a 1 nanometer spacing, each base pair effectively takes up 1.36 nanometers. This is crucial when calculating the theoretical storage density. If we imagine filling a milliliter of a DNA solution, that's a large volume. We can begin to estimate how much information we can pack in. Let's assume, for the sake of calculation, that we can arrange these base pairs efficiently, taking into account the double helix structure. A milliliter is equal to a cubic centimeter, and a cubic centimeter has a volume of 1,000 cubic centimeters. So, our volume is 1,000, 1,000, 1,000 equals 1 billion cubic nanometers. If each base pair occupies 1.36 nanometers, then we could theoretically store approximately 722.7 million base pairs in 1 milliliter. That translates into storing roughly 722.7 megabits of data per milliliter, an astounding figure. However, the reality is far more complex. The double helix structure introduces limitations. The strands twist around each other, and the density of packing is constrained. Furthermore, the stability of these base pairs isn't absolute. Quantum mechanical effects, particularly relating to the vibrational energy of the bonds between the bases, can lead to degradation over time. Temperature and pH levels in the solution also play a significant role. Even slight changes can disrupt the base pairing, introducing errors. To combat these errors, we need error correction. This involves adding redundant information, extra base pairs, to the data. Think of it like a carefully designed checksum. By meticulously monitoring these redundant signals, we can identify and correct errors that might otherwise be undetectable. Beyond simple ATGC encoding, exploring schemes where multiple base pairs represent a single bit adds further complexity, but also dramatically increases error correction capabilities. Ultimately, building robust DNA storage systems requires a delicate balance between data density and error rates. A fascinating and challenging area of scientific research. Let's explore how we actually write information onto DNA, transforming digital data into the very building blocks of life. The process begins with DNA synthesis, essentially building a DNA strand from scratch, one nucleotide at a time. This isn't done with bricks and mortar, but with incredibly precise chemical reactions. The most common method, and the one used in Illumina sequencing, relies on phosphoramidite chemistry. Think of each nucleotide, adenine, thymine, cytosine, or guanine, as a carefully packaged building block. These building blocks are attached to a growing DNA chain through a series of carefully controlled steps. The process starts with a solid support, usually a small bead, that has a short, pre-existing DNA sequence attached. Then, a protected nucleotide in the form of a phosphoramidite is added. This protected nucleotide contains the base A, T, C, or G, a phosphate group, and a temporary linker. An activator chemical then removes the protecting group, allowing the nucleotide to bond to the growing DNA chain. The activated nucleotide forms a phosphodiester bond with the last nucleotide added, extending the DNA strand. This cycle, activation, coupling, and capping, is repeated multiple times, adding the desired sequence, one base at a time. Automated synthesis machines perform these steps incredibly fast, adding dozens or even hundreds of bases in a single run. Solid-phase synthesis, as it's called, uses the bead as a platform, and the DNA strand grows directly from the bead. Quality control is absolutely crucial. After each cycle, the newly synthesized DNA is checked for accuracy, ensuring the correct base has been added and that no errors have crept in. This is done through various analytical techniques, including gel electrophoresis, which separates DNA fragments based on their size. Once the desired DNA sequence is synthesized, we need a way to read it to retrieve the information we've written. This is where DNA sequencing comes in. Next-generation sequencing, or NGS, is the dominant technology today. Illumina sequencing is a prime example. It operates on the principle of sequencing by synthesis. Imagine tiny, individual DNA strands, each representing one base. These strands are added to a reaction chamber alongside sequencing primers, short DNA sequences that initiate the process. Each base added is labeled with a fluorescent dye. As the base is incorporated into the growing DNA strand, a laser excites the fluorescent dye, and a detector captures the emitted light. The color of the light identifies which base was added. The process repeats, adding bases one by one, and the machine detects the color of the fluorescent dye at each step, ultimately determining the entire DNA sequence. The accuracy of NGS isn't perfect. Small errors can occur during the incorporation of bases. However, sophisticated computational algorithms are used to identify and correct these errors. These algorithms analyze the sequencing data and use statistical models to determine the most likely base sequence. Different sequencing platforms vary in their throughput, how many DNA fragments they can process simultaneously, and their accuracy. Newer technologies are continually improving both aspects. Comparing these techniques reveals the trade-offs. DNA synthesis can be slow and expensive, especially for large sequences. NGS, on the other hand, is dramatically faster and more cost-effective for generating vast amounts of sequencing data. The choice of method depends on the specific application, whether you're synthesizing a custom DNA sequence or analyzing the genome of an entire organism. It's a fascinating dance of chemistry and computation, allowing us to write and read information in the most fundamental way imaginable, using the very building blocks of life. Let's consider how we can ensure the information stored within DNA remains perfectly intact, even after long periods of time or exposure to various conditions. DNA, while incredibly stable, isn't immune to errors. Base substitutions, insertions, or deletions can occur during its synthesis, storage, or even when it's being read back. To combat this, we employ sophisticated error correction codes, borrowed from the field of coding theory. These codes act like a meticulously crafted backup system for our DNA data. At the heart of this system are concepts like Hamming codes. Imagine assigning extra parity bits that don't directly represent the original data, but rather act as indicators of error. For example, if we have a string of 4 DNA bases, we might add a 5th parity bit to check the overall balance. If the balance is disrupted, it signals an error. Reed-Solomon codes are another powerful tool, frequently used for correcting burst errors, where multiple bases are corrupted simultaneously. They're particularly effective when dealing with noisy environments. Let's illustrate this with a simple example. Suppose we want to store the string ATTG, representing the binary data 1010. We could add a single parity bit. The parity bit would be 1 if the number of ones in the string is odd, and 0 if it's even. In our case, there are 2 ones, so the parity bit would be 0. Therefore, the complete encoded sequence would be ATTG0. Now, let's introduce an error, a substitution where the T is replaced with an A. The sequence becomes AATT0. The error correction code, operating independently, would detect that the parity is now incorrect. It then uses the redundancy built into the system to reconstruct the original sequence. The specific method depends on the code used, but the fundamental principle remains. The extra information allows the system to identify and correct the error. The effectiveness of the code dictates how many errors it can fix. Multi-bit ECC allows for more sophisticated error detection and correction, correcting for multiple errors simultaneously. The amount of redundancy, the extra bits added, creates a trade-off. More redundancy means greater error correction capability, but it also reduces the storage capacity available for the actual data. Researchers are exploring fountain codes, which offer remarkable error correction with minimal redundancy, approaching the theoretical limit of what's achievable. These codes are incredibly efficient. Different error correction strategies exist, each with its strengths and weaknesses. Single-bit ECC is simple but less robust, while multi-bit ECC offers greater resilience. The choice depends on the anticipated error rates and the desired level of protection. Mathematical models are used to calculate the probability of successful error correction, considering factors like the error rate, the code's design, and the length of the encoded sequence. These models allow researchers to fine-tune the system for optimal performance. Ultimately, the integration of error correction codes is what unlocks the true potential of DNA as a durable and reliable storage medium, ensuring that our digital information can endure for generations to come. The careful balancing act between redundancy and capacity is a cornerstone of this fascinating technology. The very fabric of DNA, the molecule that carries our genetic instructions, is surprisingly fragile when tasked with holding data for the very long term. It's a beautiful, intricate structure, but like a delicate watercolor painting, it's vulnerable to changes over time. The core instability stems from the chemical nature of the base pairs, adenine, thymine, guanine, and cytosine, which aren't perfectly stable on their own. A process called depurination, for example, involves the gradual separation of a purine base, adenine or guanine, from its sugar phosphate backbone. Simultaneously, deamination occurs, converting cytosine to uracil, another base that can then pair incorrectly with guanine, leading to a scrambled sequence. Hydrolysis, the reaction with water, also plays a significant role, breaking down the phosphodiester bonds that hold the DNA strands together. These individual processes, occurring at a slow but relentless pace, contribute to a cascade of modifications. One particularly concerning outcome is the formation of atoxo-DG, a modified base that dramatically reduces DNA's ability to replicate accurately. This atoxo-DG is a product of oxidative damage, the attack of free radicals on the DNA molecule, often triggered by exposure to light or radiation. Oxidative damage isn't just limited to base modifications. It also leads to strand scission, effectively breaking the DNA strands themselves. Environmental factors then exacerbate these inherent vulnerabilities. Elevated temperatures accelerate chemical reactions, speeding up depurination and hydrolysis. Changes in pH, whether too acidic or too alkaline, can also disrupt the stability of the phosphodiester bonds. Humidity plays a role too, with water molecules participating in hydrolysis. And finally, exposure to radiation, UV light in particular, is a major catalyst for oxidative damage. Mathematical models, based on rate constants derived from experimental data, allow us to quantify the expected degradation rate under specific conditions. For instance, a model might predict that a DNA sample stored at 40 degrees C with high humidity would degrade by a certain percentage within a decade, largely due to cumulative base modifications. To mitigate this degradation, scientists employ various strategies. Stabilizing additives, such as trehalose, a sugar that forms hydrogen bonds with DNA, and glycerol, are frequently used to protect the strands. These additives effectively create a more stable environment for the DNA molecule. Storing DNA in inert environments, such as liquid nitrogen, minus 196 degrees C, significantly slows down the chemical processes involved in degradation. Furthermore, DNA repair mechanisms exist within cells and can be artificially induced in some cases that attempt to counteract these changes. These pathways can detect and correct mismatched base pairs or repair damaged DNA strands. However, the effectiveness of these repair mechanisms diminishes over very long timescales, and the sheer volume of damage generated continually outpaces their ability to fully restore the integrity of the DNA. The limitations of these repair pathways highlight the fundamental challenge. Even with the most sophisticated interventions, DNA's inherent instability ultimately presents a significant hurdle to achieving truly long-term data storage. It's a delicate balancing act, a constant race against time to preserve this remarkable molecule's ability to store our information. The journey to retrieve information stored within DNA, particularly after extended periods, presents a fascinating and intricate challenge. Once a sequence is written, meticulously assembled from synthetic building blocks, the preservation of that data relies on the stability of the DNA itself. However, time, combined with environmental factors, inevitably introduces alterations, fundamentally impacting the ability of standard sequencing methods to accurately read the stored information. The initial hurdle lies in the degradation of the DNA strands. Depurination, where adenine and guanine lose their hydrogen bonds, and deamination, converting cytosine to uracil, are common processes, leading to mismatched base pairings and ultimately inaccurate reads. Consequently, modifications to sequencing protocols are absolutely critical. We often employ longer read lengths. Initially, standard Illumina sequencing produced relatively short reads. Increasing the sequencing depth, the number of times each DNA fragment is sequenced, amplifies the chances of capturing a complete read, even with degraded material. This increased depth helps to overcome the lower signal-to-noise ratio that results from the presence of modified bases. Yet, even with these adjustments, the fidelity of polymerases, the enzymes responsible for copying the DNA, becomes a significant concern. These polymerases aren't perfect. They occasionally introduce errors during replication, further compounding the problem. Fortunately, several advanced sequencing technologies have emerged specifically designed to tackle these difficulties. Single-molecule sequencing, for example, allows the polymerase to work on individual DNA strands, drastically reducing errors that might arise from copying multiple degraded strands simultaneously. Nanopore sequencing, which passes a single DNA strand through a tiny pore, generates an electrical signal that changes depending on the base sequence, offering a highly accurate method for reading DNA, even when heavily modified. The challenge with retrieving information from highly degraded DNA often involves dealing with a noisy signal. The presence of modified bases creates false positives, making it difficult to distinguish between true DNA sequence and background interference. Sophisticated bioinformatics algorithms and statistical models are then employed to filter out this noise and reconstruct the original sequence as accurately as possible. Verification of DNA integrity is paramount. Techniques like terminal deoxynucleotidyl transferase, TNT, sequencing, which adds labeled nucleotides to the ends of a DNA fragment, provides a direct measure of the sequence length and can reveal the extent of degradation. Furthermore, assessing the accuracy of the sequencing data requires careful statistical analysis. Mapping the sequenced reads back to a reference genome and then quantifying the discrepancies provides a crucial metric. The goal is not simply to obtain a read, but to determine the confidence level associated with each base call. Ultimately, retrieving data from DNA stored for extended periods is a testament to the ingenuity of scientists. Combining advanced sequencing technologies with robust data analysis to unveil the information embedded within. The continued development of these technologies promises even greater precision and reliability in the future, allowing us to unlock the vast potential of DNA as a long-term data storage medium. The quest to store vast amounts of digital data within the very building blocks of life, DNA, faces a fundamental challenge. DNA, while incredibly stable, isn't immune to damage over time. One of the most promising solutions to extend the lifespan of this biological hard drive lies in a technique borrowed from the world of biology itself: cryopreservation. Essentially, we're freezing DNA to put it into a state of suspended animation, a deep slumber where degradation slows dramatically. The key to successful cryopreservation is managing the immense stresses imposed by rapid cooling. Water, a major component of DNA, behaves very differently when frozen. As temperatures plummet, water molecules begin to form intricate, damaging structures known as ice crystals. These crystals, like tiny shards of glass, can physically disrupt the DNA strands, breaking them into smaller fragments and causing significant distortions in their structure. The rapid freezing process itself generates heat, further exacerbating this issue. To mitigate these effects, we utilize cryoprotective agents, or CPAs. These substances, glycerol, trehalose, and dimethyl sulfoxide, DMSO, being prominent examples, act as shields, interrupting the formation of ice crystals. Glycerol, for instance, disrupts the hydrogen bonding network of water, making it less likely to freeze. Trehalose, a naturally occurring sugar, stabilizes DNA by interacting with its phosphate backbone, preventing the formation of damaging ice crystals and offering structural support. DMSO, a more potent solvent, facilitates the diffusion of water molecules, further discouraging ice crystal growth. The exact choice of CPA and its concentration are critical and depend heavily on the specific DNA sequence and the desired storage temperature. Mathematical models are employed to predict survival rates under different freezing conditions. These models, based on thermodynamics and molecular dynamics simulations, attempt to quantify the impact of cooling rates, CPA concentrations, and the inherent stability of the DNA. The rate at which DNA molecules are damaged during freezing is directly proportional to the rate of cooling. Therefore, slower, controlled cooling processes significantly reduce damage. A particularly slow, staged cooling protocol minimizes the size and number of ice crystals. However, the freezing process is only half the battle. The real challenge emerges during thawing. The rapid return to a higher temperature introduces its own stresses. Molecules expand as they warm, and the remnants of ice crystals can further damage the DNA. Thawing protocols must be equally controlled, often involving a gradual warming process. Sophisticated techniques like microfluidic thawing, which slowly and evenly distributes heat, are being developed to minimize this stress. Furthermore, the success of recovery hinges on the quality of the frozen sample, measured by techniques like conductivity and viscosity, and the effectiveness of the thawing process. Ultimately, cryopreservation offers a viable path towards preserving DNA data for potentially centuries, but it's a delicate operation demanding meticulous control and a deep understanding of the molecular interactions at play. The challenge of storing information within DNA isn't simply about holding the data itself. It's about managing a library of incredible complexity. Think of it like this. We've successfully encoded your favorite story into a very long strand of DNA, but that strand is now part of a truly vast collection. To find that specific story again, we need a sophisticated system for organization. This is where the digital layer comes in, the software and methodologies designed to make sense of these incredibly long sequences. At its heart, DNA indexing is the key. We need a way to quickly locate a particular sequence within this enormous collection. One common approach is sequence-based indexing, creating a digital map that links each DNA fragment to its corresponding location within the database. This map is built using algorithms that rapidly identify patterns and similarities within the DNA data. Imagine a highly detailed index for a massive encyclopedia, but instead of pages, we're dealing with millions, billions, or even trillions of DNA base pairs. Beyond simple sequencing, a critical element is robust metadata management. This means attaching detailed information about each DNA sequence, its origin, the experimental conditions under which it was created, any associated data, and even the level of degradation observed. This metadata acts as a powerful search filter, allowing researchers to narrow down the search based on these characteristics. It's like knowing not just the title of the book, but also the author, publication date, and the condition of the binding. Furthermore, efficient data compression techniques play a vital role. DNA sequences, by their nature, are highly repetitive. Algorithms can exploit these redundancies, significantly reducing the storage space required while retaining the essential information. This is akin to finding repeating patterns in a song. Instead of storing the entire song repeatedly, we can store a shorter representation of the pattern. The sheer scale of these DNA libraries necessitates powerful bioinformatics tools and algorithms. These aren't just simple search engines. They're sophisticated analytical engines capable of identifying complex patterns, predicting DNA interactions, and even reconstructing damaged sequences. They're used for tasks like identifying genetic markers, predicting protein structure, and comparing DNA from different organisms. However, managing this data presents significant challenges. Scalability, the ability of the system to handle increasing volumes of data, is paramount. Data security is also crucial, given the potential sensitivity of the information. Version control is essential, ensuring that researchers can always access and compare different iterations of the data. Finally, tracking the origin and history of each data point through data provenance is critical for verifying its accuracy and reliability. This allows researchers to understand the context in which the data was generated and to assess the potential impact of any errors. The ongoing evolution of bioinformatics is directly tied to the expanding capabilities of DNA data management. As we generate ever larger libraries, new algorithms and techniques will be developed to unlock the full potential of this incredible information storage medium. The potential of DNA as a data storage medium is a truly remarkable one, and its future impact promises to be far-reaching. Let's consider the economics involved. It's a story of significant upfront investment, but with the potential for long-term cost advantages. Currently, synthesizing DNA to store information is incredibly expensive. Each nucleotide, the building block of DNA, needs to be precisely placed, a process requiring sophisticated machinery and considerable energy. Sequencing, the act of reading that stored data back out, is also costly, though technological advancements are continually driving down the price per base pair. However, compared to the escalating costs of traditional storage media like hard drives and solid-state drives, particularly as density increases, DNA storage offers a compelling long-term proposition. Think of it like this. The initial construction of a library is expensive, but once built, the books themselves are incredibly durable and don't require constant electricity to maintain. The industries poised to benefit are diverse. Archival storage, where data needs to be kept for centuries, is a prime candidate. Imagine preserving historical records, scientific datasets, or even personal memories in a stable, resilient form. Biomedical research could revolutionize data analysis, storing and comparing genomic information from countless individuals, accelerating drug discovery and personalized medicine. Furthermore, the inherent security of DNA, the need for specialized equipment and expertise to access it, presents opportunities in data security. Encrypted data stored in DNA would be exceptionally difficult to steal or tamper with, providing a level of protection beyond current digital methods. Of course, this emerging technology isn't without significant ethical considerations. Data privacy is paramount. Who controls access to this incredibly secure data? Robust regulations and access protocols would be essential to prevent misuse. The potential for errors during synthesis and sequencing also poses a challenge. DNA is a complex molecule, and even minor changes during the writing or reading process can introduce errors. Researchers are actively developing sophisticated error correction codes, essentially redundant information built into the DNA itself, to mitigate these risks. These codes require significant computational power to decode, but they represent a critical step towards reliable long-term storage. Looking ahead, the next 10 to 20 years will likely see improvements in DNA synthesis technology, allowing for faster and cheaper production. We'll likely observe substantial gains in error correction, enabling denser data storage and increased reliability. Automation will play a key role. Robotic systems will handle the precise assembly and sequencing of DNA. Over the longer term, 20 to 50 years, it's conceivable that DNA storage will become a mainstream option, especially for applications requiring extreme durability and security. Perhaps miniaturized sequencing devices will become commonplace, allowing for on-site data retrieval. The development of entirely new data management techniques, specifically tailored to the unique properties of DNA, will also be vital. Ultimately, the future of DNA data storage is intertwined with advancements in molecular biology, information theory, and materials science. It's a story of pushing the boundaries of what's possible, a quiet revolution unfolding within the very building blocks of life itself. And while the challenges are considerable, the potential rewards, a truly enduring and secure way to preserve information, are simply too compelling to ignore. If you're still awake and enjoyed this exploration, please consider liking this video and subscribing to Ultimate Sleep Doctor. Together, we can soothe more wandering minds, one peaceful night at a time. We've journeyed through the remarkable way data is stored within the intricate folds of DNA. Let your thoughts drift, calm and unhurried, like the patterns within the strands themselves, until sleep gently finds you. Good night.